Okay, everyone. I think we'll go ahead and get started. I'm Joe Guthrie. I'm a research fellow here at Archbold. I'm the program director for the Predator Prey program, and I'm excited for everyone to be here today for an intern seminar, which is our intern, Julia Runkle. Julia is a recent graduate of Bryn Mawr College in Lancaster, Pennsylvania. Is Bryn Mawr actually in Lancaster? Excuse me for all the Bryn Mawr graduates who are joined online. I think there was 2 of you guys here at one point, weren't there? Okay, got it. Not familiar. But Julia comes to us from Pennsylvania, having previously worked at Hawk Mountain Sanctuary, as well as a couple of stints at SCA and AmeriCorps. She's going to talk today about some work she's done with data from a long-term monitoring project that Archbold and Dr. Raoul Botten have been conducting at Buck Island Ranch since about 2015. So I'm very excited to have her come up and talk to us about her work. Make sure everyone sticks around for drinks and refreshments afterwards, and we'll have time for questions at the end.
Okay, so hi. Like Joe said, my name is Julia Runkle, and today I'm going to be presenting on my independent research project studying the effects of human presence on the occupancy and activity patterns of 4 wild mammal species on a Florida cattle ranch. So before I get into the project itself, I want to briefly discuss my motivations for conducting this research. Cattle ranches contain much of the remaining habitat for native wildlife in southern Florida, and after spending some time at Archbold and Buck Island Ranch, the ecological importance of Florida ranchland really struck my interest. The mix of upland and wetland habitats found on ranchlands provide food and cover that can sustain native wildlife biodiversity. However, ranchlands require human management and activity within these habitats, and little is known about the effects of human presence on the wildlife that live in and move throughout ranchlands. So this led me to developing my overarching question for my research, which is how does human presence affect the way that wildlife interacts with ranchland habitats? So there are 2 ways in general that human presence tends to influence mammal species. One of them being habitat shifts, where a mammal will reduce its occupancy in an area where humans are present, and the other being temporal shifts, where a mammal will alter its activity patterns so that there is less overlap with human activity. So with all this in mind, I developed my research questions. I asked what relationship exists between human presence and the occupancy of each mammal species. And I also asked, does human presence influence the deal patterns of each mammal species? And then for each of these questions, I developed my hypotheses. I thought that as human presence increased, the likelihood of each mammal being present would decrease. That as human presence increased, deal activity would shift towards nocturnality. And I believe that there would be a greater temporal shift than habitat shift. Additionally, I predicted that bobcats would be most impacted by human presence, followed by raccoons and deer, and that wild pigs would be least impacted. So now getting into how I went about creating a study to hopefully answer these questions. I chose to focus my study on Buck Island Ranch as it provides the unique opportunity to do research and collect data on a working cattle ranch. Furthermore, it consists of 10,500 acres with habitats including improved pasture, semi-native pasture, oak and palm hammocks, as well as wetlands. I chose to focus on the year 2016, and I look at a full year of data. The species I studied were raccoons, bobcats, wild pig, and white-tailed deer, and all of the data came from game cameras. The game cameras, if you look at the map on the right side of the screen, are highlighted in blue, and they're in 1 kilometer square grid cells all across Buck Island Ranch. And then on the top right of that image, there's a kind of dark image of one of our cameras. And these cameras take 10 photos per trigger and have a 5-minute quiet period after each trigger. The data from Buck Island Ranch goes through AI processing in order to identify species within images. And the way that the AI is set up, there are several species categories that refer to humans. These categories include images of researchers, hunters, and cowboys walking and on horseback, vehicles, and domestic dogs. It's assumed that these dogs are accompanied by a human or otherwise related to human activity. And throughout the rest of this presentation, I will be referring to all of these categories as humans. So I've been throwing around a lot of words like human presence, deal activity, and detection, but what do they mean in the context of my project? Before I continue with my study overview, I will define this important terminology. The first thing I'm going to talk about is human presence, which I divided into two subcategories: human occupancy, meaning whether a human was present or absent at the camera site that day, and human abundance, which I defined as the number of people detected per camera per day. I also broke human abundance down further into human abundance by location, meaning the number of people detected per camera over a full year of data collection, and human detection or human abundance by date, meaning the number of humans detected per day across all camera sites. I use these same definitions for mammal presence, occupancy, and abundance. For deal activity, I use the definition of a density of detections over a 24-hour period. And for detection, I defined as a photo of the species at least 30 minutes after the previous photo of the same species. So in order to make sure that any relationship between human presence and mammal occupancy was truly caused by human presence, I chose to include a number of covariates that I thought might influence this relationship in my study. These covariates include trail type, habitat, pasture type, season, and breeding season. And then I have the factors that I included within each of those covariates listed on the left, and the breeding season of each species highlighted in the bottom right corner. So before I even went about doing anything with my data, I wanted to get a little bit of summary statistics. So I found the total number of detections of each species with 2,952 humans, 5,969 raccoons, 1,093 bobcats, 7,053 wild pigs, and 7,147 white-tailed deer. So I spent a lot of the time on my project doing data management, and I know that that can be pretty boring to have to listen to, so I'll try to keep it short and just highlight a few key steps. As I mentioned earlier, the cameras would take 10 pictures per trigger, so each detection would consist of 1 to 10 photos. And in order to have cleaner data frames, I made each photo sequence 1 to 10 rows of data per detection into 1 row of data per detection. I then made rows for each 30-minute interval of every day of the year with a column for presence or absence of each species regarding that 30-minute interval. I then added a column for each of my covariates, and I realized that there were a huge number of 30-minute periods with 0 animals and 0 humans present, and that this actually ended up skewing the data. So I derived my final data set to only include rows where at least 1 mammal or human was present. I could then summarize the data by the sum of the number of detections per day to get human and mammal occupancy, as well as human and mammal abundance per location per day. Or I could use the unsummarized data to get human and mammal abundance by location and human and mammal abundance by date. So the first thing I wanted to look at once I had all of my data together was the detections across camera sites. I used the number of mammal detections per location in order to understand which camera sites had the most detections of each species in 2016. So before I get into this map in particular, I just wanted to briefly describe what the next few maps are going to look like. The numbers on the map represent the camera numbers, and the size of the bubble represents the number of mammal detections at each site. This one here in particular is looking at raccoon detections on Buck Island Ranch. There was a minimum of 3 detections at a camera site and a maximum of 557, and the most detections occurred at sites 16, 10, 35, 36, 37, and 38. And all of the areas with the most detections were in semi-native pasture, and they were in the East and South Marsh. This is looking at bobcat detections. There was a minimum of 1 detection at a camera site and a maximum of 130, and the most detections occurred at sites 8, 37, and 38. And these are also in semi-native pastures in the East and South Marsh. Wild pigs had a minimum of 13 detections at a camera site and a maximum of 762, and the most detections occurred at 16, 27, and 40. And these are also in semi-native pasture type in the East and South Marsh. And white-tailed deer were more evenly dispersed across sites than other mammal species, with a minimum of 16 detections at a camera site and a maximum of 497. And finally, this shows the human detections on Buck Island Ranch for 2016. There was a minimum of 14 detections at a camera site and a maximum of 589, and the most detections occurred at sites 38, 25, and 27, and all of these are located on main access roads. The next thing I wanted to look at once I had all of my data together was mammal species daily activity patterns. In order to look at the changes in daily activity patterns of each mammal species with human presence, it was first important to understand the normal deal activity of each species regardless of human presence. And so the following graphs will show the density of activity across time over a 24-hour period. These graphs give a quick comparison of human activity and the activity patterns of each mammal species to show the overlap in activity regardless of human presence. These graphs are centered at noon, and the yellow bars represent sunrise and sunset. The dark blue lines represent the mammals' daily activity patterns, and the humans are highlighted in the orange lines. As you can see from these graphs, the mammal species already tend to be more nocturnal or crepuscular regardless of human occupancy, and therefore their activity patterns already have little overlap with human activity patterns. In terms of statistical analysis, I performed two main tests, one being binary logistic regression modeling, which I used to understand the significance of covariates, both separately and with interaction between each covariate and human abundance, in terms of their impacts on mammal occupancy for each species. The other test I used was kernel density overlap, which I used to understand if there was a significant difference in the deal patterns of each mammal species between different levels of human abundance. So now we can get into some of the results from my project. I ran a binary logistic regression model to understand the relationship between the number of mammal detections at low and high human abundance. I defined high and low human abundance based on the number of human detections at each camera site from the whole year of data, and I separated them by the median number of human detections per camera, with 0 to 37 being low human abundance and 38 or more being high human abundance. And then I also created these graphs to help visualize this relationship. As a reminder, the numbers on the map represent the camera numbers, and the size of the bubble represents the number of detections of the species at each camera site. This here is showing raccoon detections at low and high human abundance camera sites. The cameras with the most raccoon detections seem to be at camera sites with low human abundance, such as 16 and 10. However, there are still many detections at camera sites with high human abundance, such as 35 and 38, which are still rather large bubbles. And the binary logistic regression model showed that any difference between high and low human abundance was insignificant. The cameras with the most bobcat detections seem to be at camera sites with low human abundance as well, at sites 8 and 37. However, there were still many detections at cameras with high human abundance, such as 38. And the binary logistic regression model again showed that these differences were not statistically significant. The camera with the most wild pig detections seemed to be at a camera site with low human abundance at camera site 16. However, sites 27 and 40 at high human abundance areas also had a large number of wild pig detections, and the binary logistic regression showed that any difference is not statistically significant. And finally, the number of white-tailed deer detections are relatively evenly distributed across low and high human abundance camera sites, and the binary logistic regression model confirmed that any difference between the number of detections at high and low human abundance was statistically insignificant. So just to summarize, although areas with high human abundance seem to have lower bobcat and raccoon abundance, the areas with low human abundance, the difference was not statistically significant. This graph illustrates the binary logistic regression model showing the relationship between the number of humans detected per camera per day and the occupancy of each mammal species per camera per day. The effect of human occupancy on species occupancy for all of the mammal species was not significant. However, the effect of human abundance was significant for all of the mammal species. As you can see in these graphs, the effect of human abundance has a negative relationship with species occupancy for all of the mammal species. For raccoons, wild pig, and white-tailed deer, the species occupancy declined with an increased number of human detections, and species occupancy reached 0 at around 5 human detections per day. For bobcats, species occupancy had a steeper decline, and species occupancy reached 0 at around 3 human detections per day. The following graphs depict the binary linear regression models that I ran to understand the effect of each covariate with interaction on the relationship between the number of human detections per camera per day and the occupancy of each mammal species per camera per day. When there was no human occupancy, the mammal species utilized all trail types, but utilized unimproved roads slightly less than other trail types. When humans did occupy the landscape for all species to different degrees, unimproved road had greater species occupancy regardless of the number of human detections. For wild pigs, cow trail also had greater occupancy than open or canal trail types, but less than improved or unimproved road when humans were present. This graph is looking at the covariate of habitat. When humans were not present, wild pigs and wild deer had similar occupancy in all habitat types. Raccoons had higher occupancy in wetland habitat types than pasture or hammock, and bobcats had greater occupancy in hammock and wetland habitats than pasture habitat types. However, when humans were present, all species had greater occupancy in pasture habitat than wetland or hammock habitat, regardless of the number of human detections. These graphs are all looking at pasture type. While all species had a significant positive relationship with semi-native pasture type in relation to species occupancy, when the interaction between the number of human detections and pasture type were considered, pasture type did not have a significant effect on raccoons, bobcats, or deer. Furthermore, semi-native pasture type had a significant negative relationship with wild pig abundance when interaction was considered. In other words, pigs had greater occupancy in semi-native pasture when humans were not present, but pigs had greater occupancy in improved pasture when the number of human detections increased. This is looking at the covariate season. When humans were not present, white-tailed deer and wild pigs had similar occupancy during the wet and dry season. Raccoons had greater occupancy in the dry season whether humans were present or not, and regardless of the number of detections. And bobcats had greater occupancy in wet season than dry season when humans were not present. However, when humans were present, all species had greater occupancy in the dry season. And this was regardless of the number of human detections. And finally, looking at breeding season, white-tailed deer were the only species where breeding season had a significant effect on their occupancy. When humans were not present, occupancy was similar between breeding season and non-breeding season months. But when humans were present, the probability of white-tailed deer occupancy was greater during the breeding season than the months that were not during the breeding season. And I have the months of the breeding season highlighted in the bottom corner. So just to go over all the graphs we just saw, human occupancy did not have a significant effect on the species occupancy of any of the mammal species. However, human abundance did have a significant effect. An increase in the number of human detections per day significantly reduced the occupancy probability of each mammal species, and this trend was most pronounced for bobcats. In terms of the other covariates, while they influenced the relationship between human abundance and mammal occupancy, they did not explain this relationship. So these graphs are looking at deal activity for each mammal species on days with high and low human abundance, with low human abundance being light blue and high human abundance being dark blue. And then the overlap is in the bottom corner there. So these graphs are centered at midnight, as the species in the study tend to be nocturnal or crepuscular. And the yellow bars represent sunset and sunrise. For raccoons, during days with high human abundance, there were two distinct peaks in the density of raccoon activity, one being right after sunset and a smaller one right before sunrise. However, on days with low human abundance, these peaks were less distinct and the density of activity remained more constant throughout the night. And the overlap between the two activity patterns was around 95%. For bobcats, during days with high human abundance, there were two distinct peaks as well in the activity, one right after sunset and one right around sunrise. On days with low human abundance, the peaks shifted with a peak right around midnight and a peak right before sunrise, and the overlap was around 86%. For wild pigs, during days with high human abundance, the density of wild pig activity was highest at night, with a slight gradual decrease from sunset to sunrise. On days with low human abundance, the density of activity peaked after sunset, with smaller peaks right after midnight and right after sunrise. And the overlap between the two activity patterns was around 93%. For white-tailed deer, during days with high human abundance, the density of deer activity had high peaks right around sunset and sunrise, and low activity during the days and nights. On days with low human abundance, the peaks were slightly lower at sunset and sunrise, and there was a small peak right around midnight, and the overlap between the two activity patterns was around 90%. So from these graphs, it appears that at high human abundance, raccoons, bobcats, and deer tended towards more crepuscular activity, whereas at low human abundance, they tended towards nocturnality. And the opposite seemed to be the case for wild pigs, but the shift was less pronounced. All the species still had relatively high overlap between low and high human abundance, and this is perhaps due to the low overlap between mammal species and human activity patterns in general that we saw from the first deal charts that I showed earlier. So now getting into the discussion and getting back to the main question for my research. How does human presence affect the way that wildlife interacts with ranchland habitats? Well, in the short term, I found an increase in the number of humans per day significantly reduced the occupancy of each species at the camera site that day. Additionally, many of the mammal species shifted from nocturnal peaks in activity to crepuscular peaks in activity with higher human abundance. In the long term, or the span of a year, mammal species did not avoid areas of Buck Island Ranch exhibiting higher human abundance. So overall, based on this study, human presence appears to influence how wildlife interacts with ranchland habitats in terms of both habitat shifts and temporal shifts in the short term, but it does not have a significant effect on wildlife in the long term. However, more research is necessary to fully understand the complex responses of wildlife to human presence within ranchlands. In terms of these future studies, I had a couple of ideas. The first idea I had was using other methods of detecting human presence. I think it would be really interesting to use ARUs or acoustic recording units to see how noises associated with humans might influence mammal occupancy and activity. Additionally, all of Archbold's cameras and a few of the ranch cameras already have these units. I also thought that the use of a multi-species occupancy model could be used to test interactions between species to see if human presence affects species interactions and see if there were any changes in predator-prey interactions with high human abundance. A longer time scale could be used to collect more data, which might help to clarify changes in species deal activity patterns with human abundance. And finally, I thought that looking at additional sites might be helpful. We could utilize these methods to look at a ranch with greater human presence and compare with Buck Island Ranch to see if the same trends are found, or we could also compare with conservation lands such as Archbold or the FWC WIA. And before I conclude, I just wanted to give a special thank you to Joe Guthrie, Raoul Boughton, Emily Angel, Finn Olween, previous predator-prey interns Gabriel and Shriram, all Archbold interns and Archbold staff. This has been an incredible opportunity, and I just want to thank everyone here as well, if you're not listed above. And it's important that I note that the data was provided by the USDA APHIS Feral Swine Management Project, which is led by Dr. Raoul Boughton. Anyone have any questions? So people can hear you online.
Thank you. Yep. What made you choose the species that you did?
So I chose to look at these species because I wanted to have a diversity of mammals. I wanted to look at a predator species, a common game species, and an invasive species to see if there were any differences between each of the species. And I also wanted to have more of the common species so that I could have more data for effective statistical analysis.
Thank you. Thanks. That was a great presentation.
Thank
Loved
you.
it. Amazing graphs. Also like all your animals. But it seems that you found there are greater impacts of human activity on bobcats more than some other
Yes.
species. I'm wondering if you could talk about a little bit more about the biology of some of the species and why humans have different impacts on the different species that you focused on.
Yeah, the different species require different habitat types, and they have different things that they eat, I guess. So the wild pig and the deer might be actually have a positive influence with human abundance. It wasn't shown in any of the graphs or any of the research that I did, but with more human related food sources, such as the food for cows and stuff on the ranch, that might actually cause them to come into contact with humans more often and be more occupy the areas with human presence. And that's not necessarily the case for bobcats. They would not necessarily be attracted by human presence. So that might be a cause, or there might also be other covariates that I didn't look into that could have caused bobcats to not occupy the areas with human presence.
I guess that kind of answered my question, but I was thinking specifically for the wild hogs, like why were they having an interaction or like a positive correlation with human presence in the improved pastures, but not the semi-native pastures?
Yeah, I'm not
Yeah.
entirely sure why, but I would guess that it has something to do with the cattle food source.
Hi, Julia. That was a great presentation. So it looks like this is a positive plug for ranchlands as a place where humans and wildlife can coexist potentially. But my question was, uh, where is the cutoff? So if you don't find significant differences, where do you think, or why do you think that is? Like what would increase in human activity actually show the trends that you predict?
I'm not sure if it would or would not show. I think more research is necessary to come to a definite conclusion on that. I think that there is a lot of the ranch land in the area is like the only habitat that a lot of these species have, or it's the majority of the habitat that they have at their disposal to use. And so that might be a reason why some of the species are seen there, even when there is human abundance. But it would definitely be interesting to see at areas with higher human activity to see if these trends continue.
Yeah, it was a great presentation. I had a question because so it looked like the timing of things. It looks like in the absence of humans, most or maybe it was one of the species in particular like to be out and about in the middle of the night. But then when there were humans, then they moved towards the crepuscular hours, and that seems counterintuitive to me. But is there like a good reason? Are they not afraid of other predators, and so they move out?
Yeah, I found that really interesting. That I think was bobcats. I can try to find the slide. But it stopped my screen sharing. The bobcats showed more crepuscular activity than nocturnal activity, and this went against what my prediction was. I don't have any reasons from the study for this to occur. I think that other covariates could definitely be involved, but I'm not sure why that would occur.
Hi.
Hi.
I should have raised my hand. Great presentation. I'm wondering, I was surprised at the number of bobcat observations. It seemed like a lot. And I'm wondering in this field, is there a way to use number of observations to predict population size?
I think there is. I know it's usually easier with like DNA collection, but if there are enough differences between the species, like the individuals within the species, cameras can be used, especially if ear tags or something like that is deployed. And then from there, population densities could be guessed at or found.
So how big do you think the bobcat population is at Buck Island Ranch?
I'm not sure. My study didn't take into account if the same individual was passing by the camera like 27 times, or if it was different individuals each time. And so it's hard to get from the numbers that I had in my study to like actual population estimates.
Passing the mic
Yeah.
down.
Great talk. I was just wondering, since the species and humans didn't have a lot of direct interactions, what it is exactly that's kind of cluing off these species to humans, if it's like fragrance or disturbance or something else.
Yeah, I am not sure. That's one of the things that I think would be really interesting to look into, especially with the acoustic recording units, to see if it's the noise that humans create that was cluing off these animals that humans were present and that there was like potential danger. But I cannot be sure from the study that I conducted.
Thank you.
Okay, this question comes from Josh Daskin. Very nice presentation, Julia. For occupancy, the 30-minute break between detections makes some sense. Would there be any interesting additional questions, e.g. about frequency of species presence, that might be answerable from not discarding any detections?
Yeah, I think that it could potentially be helpful to look at the data in its raw form with just 5-minute quiet periods between each detection. I chose to have a 30-minute difference to better look at deal activity. And one of the things I had wanted to look at was nocturnality, but I didn't include that in my presentation. And so
Nocturnality is the tendency to be
shift
active at night?
Yeah.
Okay.
I didn't look at it in as much detail as I would have liked in my presentation, but that's the reason that I chose the 30-minute interval between detections. But I do think that just having that 5-minute difference could be helpful in seeing if there were differences in detections. Yeah.
So I was just wondering about, obviously you've got the measure of human activity as people passing by. Another measure would be sort of animal use days in each of those pastures, maybe, because we've got the sort of extra factor is just the presence of herds of animals in those pastures may alter it. So I wondered if you'd given any thought to using those animal use days by pasture versus the camera totals, and whether to crosswalk the human activity with the animal use days. I presume to some extent they're strongly correlated.
Yeah, I definitely think that would be interesting to look into. I know Gabriel, one of the previous interns here, looked at bobcat activity with cattle use, and he was mostly using the camera data. But it would also be really interesting to look at those animal use days to see if there is an impact on the occupancy of mammal species, because I think that there is a large correlation between those days and human activity on the ranch.
Raul.
You want a hard question now?
No.
Because I know the data really well. So I've got a question. We've talked about it, but I think it's good for people to know this part of it. So we talked about low, or you've talked about low and high, human presence, and you used a binary cut there, or used abundance.
Mm-hmm.
So what was the distribution of low and high use across the year? And
So,
how could that affect sort of what you're talking about?
Yes, it was definitely different throughout the year. There was a lot higher human abundance in the winter months and during the dry season. And I tried to account for that in my deal charts with the difference between low and high human abundance. I split the data so that I had summer data and I also had winter data. And then I looked at each of them with high and low human abundance to see if there were differences and if those differences were caused by the seasons. And I didn't find a significant difference in the graphs between the two seasons. So the graphs that I showed were the only included data from the winter months.
Okay, I think that about covers it. So you've got all your questions out. Well, we have one more round of applause for Julie.